and she is presently director of IB Carb and Carbomet Networks and a spin-out company, ByShape. I welcome her to chair this session and conduct this session. Thank you.
Thank you very much. And welcome to this session. And our first speaker, as you can see, is already here. He's Sriram Subramaniam, and I'm very pleased to introduce him for the first lecture because Sriram and I overlapped at MIT in, I think, 1988, I think it was. And actually, Sriram was the one who taught me quite a lot about lipid biochemistry, so I'm forever grateful to him about that. So Sriram, just shortly his biography. Take my glasses off here. So he got his PhD in physical chemistry at Stanford. He did a postdoc then with Gobind at MIT, and he's now the chief of biophysics section at the Center for Cancer Research at the National Institute, or Cancer Institute USA, and also visiting faculty at Johns Hopkins. His interest is in the development of advanced technology in imaging using 3D electron microscopy, a very hot topic. And he applies this to HIV, AIDS, and cancer research. And we're looking forward to his talk. Sriram, go ahead.
Thank you, Sabina. It's very good to see you after all these years, and I'm really excited to have an opportunity to share this work with the audience. I'm going to switch from my personal video to sharing my screen. Hopefully this will work well. Please let me know if it doesn't work. Can you now see my screen? Okay. Okay, I'd like to share with you work we've been doing in my lab in the last 1 or 2 years. And I'd like to begin with remembering my time in the Purana Rajbhandari lab. I joined about 30 years ago, 30 years and about 7 months ago in April of 1987. I stayed there for 4.5 years. I left in October of 1991. During this time, I presented 16 one-day seminars. Many of you will know and remember these very well. I presented 10 Wednesday seminars and 12 Friday Journal Club seminars. And this is from a time when I came, when I did my PhD at Stanford. In the entire time I was there, we had 4 group meetings in the 5 years that I was there. So it was a change of pace for me. But the most important thing is the length of impact continues unabated. And I remember almost every day the time that I spent with Gobind. And so I'd like to thank them, and Tom in particular. Unknown to you, you've been a major influence in many of the things I did then and continue to do. So thank you. I'd like to present my talk in 4 sections. I'd like to first introduce the cryo-EM, which is now, as Sabine said, become rather a hot topic. I'd like to give 3 sets of examples of work in my lab. Application to determination of structures of dynamic metabolic protein complexes, work on structural studies of ion channels, membrane proteins, and finally applications to one example of a nucleic acid protein complex. Cryo-EM is hot as attested by many things, including the Nobel Prize this year, but in January 2016, methods proclaimed cryo-EM to be the method of the year and showed the structure of beta-galactosidase, which we had reported at near close to 2 times resolution in 2015, superposed on the melting polar ice cap. And the point was that these methods were being increasingly used, and our work had shown that you can actually get essentially atomic resolution with cryo-EM. One of the changes in cryo-EM the last decade is the ability to investigate structures of smaller things. Cryo-EM as a discipline itself is not new. It's been used by a number of scientists in the lab for well over a decade. But while 10 years ago, we could investigate the structures of the large entities, like icosahedral viruses, the large protein complexes, such as ribosomes or proteasomes, in more recent work, the fact that we can now extend this to look at much smaller and especially dynamic protein complexes, it makes it very exciting for a very broad range of problems of interest in cell biology. I will mainly speak about some of these, the work on these smaller complexes, which is where most of my own focus has been. And I'd like to begin with a brief video that introduces the method to those of you that may not be familiar with how it works. On the face of it, it looks very easy. Essentially, it begins with a purified protein or purified virus or whole cells in many applications. This is delivered the surface of an electron microscope grid that's about 3 millimeters across, plotted with Wattman #1 filter paper that most organic chemistry labs have. And at this point, the protein or the suspension is in an aqueous buffer, which is sponge frozen in liquid ethane that's cooled by liquid nitrogen. At this point, we vitrify the sample, we freeze it in its tracks, and it can then be delivered into the column of an electron microscope via a cassette, which is also cooled to liquid nitrogen temperature. Once it goes into the column of an electron microscope, we can irradiate this with an electron beam, collect images that are essentially projection images of these interventions. So each of these structured things you see here are images of the protein, and one can then computationally process these images to reconstruct the 3D structure. So many of these data sets are somewhat automated, and I think the excitement is that methods can now especially be very generally used. Although we've been working on cryo-EM for some time with the development of new detectors, we essentially showed in 2015, that's 2 years ago, that one could begin to use cryo-EM methods go past what many of us thought would be a sound barrier for cryo-EM technology, which is about 3.5 angstrom. We showed that we could get to these really high resolutions where you can actually visualize densities with side chains directly. So at these kinds of resolutions, for those of you who are used to looking at crystallographic density map, this is essentially an EM density map, where you can visualize densities for side chains directly. You can place them into the density map. And I think the fact that we can now visualize unambiguously these side chains is very exciting. In addition to visualizing the side chains, for example, with aromatic rings, we could clearly visualize fold in aromatic residues, which typically are associated with structures at resolution of angstroms or thereabouts. But perhaps the most exciting finding was that we could visualize density water molecules. The idea that we could visualize water molecules in a cryo-EM structure was actually very surprising to us and, of course, transformative. Because at these kinds at resolution that you can see water molecules, it becomes possible to now look at chemistry of the structure itself as well as chemistry of small molecules bound to the protein. And this was my main interest, which was to begin to localize the binding sites of small molecules that actually bind to the protein. In this case, G is a molecule that inhibits beta-galactosidase. And not only could we show that it was bound, we could also show density for a single water molecule between PETG and the active site glutamic acid residue. Last year, we extended this to real-life applications, and this was always the goal. And in work we did with a cancer target, a AAA ATPase called 297. We demonstrated that we could use cryo-EM methods to achieve high-resolution structure for P97, not only by itself, but in complex with a molecule being developed at the National Cancer Institute as a possible inhibitor of this enzyme. So this was the very last year when this paper was published. This was the first example of the application of IEM to a clinical target, and also the first example where we showed that a small molecule could be visualized at essentially atomic resolution when bound to a protein target such as P97. The reason this was interesting technically is that P97 has been known for a long time. It's a very important protein. It's involved in protein degradation, it's involved in autophagy, and many other important roles. And over a decade ago, work at Stanford has shown that uh, the structure of the protein in the ADP bound state, adenosine diphosphate, the structure of the protein is it's a hexamer, and it has these three domains that you see colored on the right. The central question in the field was the nature of the conformation changes that take place when ADP is exchanged for ATP. And this remained structurally a puzzle because it was almost impossible to crystallize the protein in terms of ATP. And although there were many structures reported with analogs, it was clear that the changes were not being captured in the crystallographic studies. And we showed last year in this paper that I briefly mentioned before that we now understood the origin of this problem. And this is demonstrated in this model, which shows structure in the ADP bound state. And what we showed is that when the first molecule of ADP is exchanged with ATP gamma S, there's a change in one of the nucleotide binding domains that's shown in magenta. But when the second molecule is exchanged, there's a much larger structural change, leading to an opening out of the N-terminal. Now, the reason why it's difficult to see all of this with crystallography is these three conformations are present simultaneously in solution. And we showed that it is possible to computationally separate three conformations and solve each of them at atomic resolution. So now we have these three structures, each done at atomic resolution. But our interest was, in addition to figuring out the mechanism, to be able to map the molecules at high resolution. And the molecule you see up on the upper left, the phenylindole derivative, that at that time had been shown to have specific activity against P97 ATPase, at least the P97 itself, versus other ATPases. But we got very lucky in the sense that we demonstrated the binding of this molecule freezes the protein in its tracks, and allowed us to determine, as I said before, the very first high-resolution structure by cryo-EM of a small molecule bound to a protein target, and begin to get information on the detailed interactions in the binding site. This is the kind of thing that crystallographers have been doing for 2 decades or more, but it's exciting that now we can get to this level of information with cryo-EM technology. We continued this work last year, trying to further advance the boundaries of resolution that we achieved with cryo-EM, and also to begin to push the boundary of these sizes. We always wondered, would cryo-EM be acceptable to look at smaller proteins? In this paper there, where we reported advances on both fronts, we took on a set of proteins which are dehydrogenases, glutamate dehydrogenase, isocitrate dehydrogenase, and lactate dehydrogenase, that's in the middle. And the transition from glutamate to lactate to isocitrate dehydrogenase essentially is is a progression in size that gets smaller and smaller. And IDH, isocitrate dehydrogenase one in particular, is now below 100 kilodaltons. And this was a huge milestone for us to get to. Because if you look at this metabolic pathways chart that almost all of you are familiar with, actually used to be in the corridors of Govind's lab at MIT, is where I saw it for the first time. The goal really is to have these metabolic pathways come alive in 3D, because although we know the identities of many of the molecules in this chart, we also like to know what this looks like in 3D. That drove much of the work in my lab for a decade. But now in the context of cancer and other diseases, where many of these proteins are affected or mutated, what we seek to understand now is the way in which these molecules might inhibit specific enzymes in various aspects, involved in various aspects of metabolism. This work, which we reported last year in Cell, also enabled us to push the advanced resolution achieved by cryo-EM, where we showed that glutamate dehydrogenase could now be resolved at 1.8 angstrom resolution, and this still today is the highest resolution structure reported by cryo-EM in the electron microscopy data bank. At this resolution, you can see densities of side chains, as I showed before, as well as densities on water molecules, many of them that you see circled here. The more important thing we were after from a clinical perspective was to demonstrate that we could visualize a small molecule being developed at the NIH called ML309 that specifically targets a mutant form of isocitrate dehydrogenase. And the reason this is important is that patients that have mutations at this particular residue have cancer that is due to the change in metabolism. So instead of isocitrate producing alpha ketoglutamate, which is what it does in normal cells, in the case of patients that carry this mutation, it in turn produces 2-hydroxyglutamate, which is an oncometabolite. So the task of finding molecules that inhibit IDH1 is to find ones that specifically inhibit the mutant form, but not the wild type form. And there has been success on these things, including success at the NIH, as well as two other commercially reported commercial entities that have reported success in molecules that block IDH1. Now, for the first time, we show that we can use cryo-EM methods to go after locating a small molecule in IDH1. And it's significant because this has not yielded to crystallographic analysis for reasons, the same reasons as with 97. Once the molecule binds, it's in equilibrium between two states and it's present at the same time. You can now computate these up. So this also demonstrated that cryo-EM could now be used to study proteins smaller than 100 kilodaltons, which many of us thought even last year would be almost impossible. The application to membrane proteins, which I learned about really for the first time in MIT, has also been very interesting, and worldwide there is tremendous interest in applying these methods to study membrane proteins, in particular because they can be challenging to study by crystallography. Among the proteins we chose to work on, we focused on ion channel. And one example of this is work on ionotropic glutamate receptors, work that we did with Mark Mayer at the NIH. And as many of you will know, these are critical proteins in the brain. When they don't work properly, it can lead to diseases such as Alzheimer's, memory loss, Parkinson's, and many others. And there is therefore great commercial interest in finding molecules that might target one or the other subtype of these ion channels. Eric Guo, who also was in Govind's lab about the time that I was there, reported a seminal piece of work in 2008 or 2009, where he showed the structure of the resting state, the AMPA receptor. And from that point onward, the challenge to the field for the next decade was to decipher the nature of the gating cycle. What actually happens when glutamate binds, and how is this relevant to the transport of ions across the membrane? And in a series of papers that we published along with Mark Mayer, first in 2014, and then second in 2016, we demonstrated that using cryo-EM methods, we could determine the nature of structural changes that resulted from glutamate binding to activate the receptor. But even more interestingly, we also demonstrate the changes that took place with desensitization of the receptor. These two processes of activation and desensitization are processes that happen in milliseconds. And understanding their structural basis is, of course, critical to drug discovery. And the work we reported last year is also technically significant because not only is it the first, and to my knowledge, the only atomic resolution model for the desensitized state of a glutamate receptor, we also showed that we could visualize densities for carbohydrates that you see on the lower right. Each of those are end-linked sugars that we can visualize density for directly close to the protein. And unlike crystallographic analyses, we don't need to truncate these residues, or to truncate the sugars or mutate the residues, because we're just averaging the structures of the molecules. And if it's disordered, it doesn't get averaged. And whatever is ordered and close to the asparagine residue shows up in the density map. Among the other things that showed up in these studies was a very interesting and surprising finding that I'd like to share with you here, which is the discovery that desensitization leads to a very large structural change. We all know examples of allosteric proteins with very large changes. But what was initially puzzling about this was the fact that typical cross-linking studies had shown that the same residues were cross-linked in the resting state and desensitized state. And yet, we were finding a very large structural change. And it turns out the answer to this puzzle is that the changes are so large that the same residues are cross-linked, but in the neighboring subunits. So this is a tetramer. And it turns out the changes are so large, and the rotation is to an extent that it actually leads to a contact of the same residue one subunit over. And of course, that's why the cross-linking study suggested the same residues were cross-linked, but it did not mean that there was no structural change. In similar studies, we worked recently on an ion channel that transports magnesium. This is a prokaryotic ion channel. It's called Core A. And this is work we did with Eduardo Peroso at the University of Chicago. And the reason we were interested in this ion channel is because crystallographic studies had shown for several years that the structure of the protein was independent of the presence of magnesium. So the structures that you see superposed are with and without magnesium. And the surprise here to biochemists and electrophysiologists is that the changes that people observed with spin labeling studies and with other readouts implied that the magnesium binding or loss of magnesium seemed to change the protein structure a lot, but this didn't show up in the crystallographic analyses. So our guess was that the crystal structure was essentially inhibiting the true structural change. And to do this, we essentially initiated intensive collaboration with Eduardo Peroso to test the structure with cryo-EM in the presence and absence of magnesium. And our first results were to demonstrate the structure with magnesium, and indeed it showed that the structure was the same as the crystal structures. We could visualize density of side chains, magnesium ions. It was also a technical advance for us at that time last year, because we showed that now proteins in the size range of 200 kilodaltons, membrane protein, could be tackled with cryo-EM. The exciting discovery came when we took magnesium away. What we found, to our great surprise, was that in the absence of magnesium, the protein undergoes a very large structural change. We're talking now changes of the order of 30 angstroms that are associated with loss of symmetry. So not only does the protein change a lot in structure, there is no longer five-fold symmetry. Ever since Rod McKinnon's work, many of we were conditioned to think of symmetric opening and closing events in ion channels, or perhaps small changes. But now that we've removed the constraints of a crystal lattice, I think it could be a paradigm shift in terms of extent of change that these ion channels display when they're in the context of a membrane, but without the context of a 3D crystal. The last example I'd like to share with you is work just published on the application of cryo-EM to nucleic acid protein complexes. And this work we did in close collaboration with Dinshaw Patel, who is at the Memorial Sloan Kettering Center at New York. And on the left, the schematic on the left-hand side essentially captures the structural biological problem we are trying to solve, and the biology that's relevant to this. When bacteria get infected by bacteriophages, uh, CRISPR complexes, which are now very much in the news, swing into action, and one of two outcomes is possible. Either the DNA is recognized by a CRISPR complex, and it leads to cleavage of the DNA, and the bacterium survives, or the bacterium is destroyed, and it leads to essentially it's a replication of the phage. What we showed is in a structure that actually was perhaps the most challenging we've done. The near atomic resolution model for the CRISPR-Cas complex, 9 polypeptides, no symmetry, with RNA and DNA. And what emerged from this work again is the revelation of the extraordinarily dynamic nature of this complex. And what we've shown now is that the binding of DNA leads to this dramatic change in phase. Essentially, you can think of these complexes as being in a quiescent state until DNA arrives. And when the right DNA arrives, it's recognized by the PAM motif, it opens out the structure and then leads to a nuclease that cleaves the DNA. We now have structures with numerous inhibitors that compete with this degradation, the phage fighting back, and now we have an understanding of the location at which these inhibitors bind. This is one such example, an inhibitor called ACR10 that was reported by Alan Davidson at Toronto. And we show that the mechanism by which this inhibitor inhibits phage, the function, is to sit at exactly the same side. The DNA binds, you see this superposition on the left, and also it leads to interesting structural changes and to essentially a clasping of the inhibitor by movement of the hook domain that you see on the right. Essentially, now this opens the door for us to look at not just the effective DNA binding, but also the different ways in which inhibitors block DNA binding and thereby enable the phage to replicate. So these are 3 examples I've discussed with you. Work on the one hand, the metabolic protein complexes, ion channels, and nucleic acid protein complexes. And the foundation for essentially all of these example studies was laid in my time at MIT, where I was exposed to the very rich biological landscape of what is possible. And I'm, as I said before, really grateful that I had this opportunity. The cryo-EM field itself has now evolved from a time 15 years ago when, actually 20 years ago, when I did the very first structure of beta-galactosidase with Richard Henderson, and when we had 30 action resolution at the left, but we are now at essentially atomic resolution on the right side. And I want to end by pointing out that as the field grows, more and more structures are being done at high resolution. This plot shows you where the field is today. The x-axis is resolution achieved. The y-axis is size of protein. And you can see that the area which is really most interesting from a therapeutic point of view for drug discovery, we believe is to be able to look at small proteins at high resolution. This is the lower left corner. There's about a dozen entries now at high resolution by cryo-EM. I've spoken to you about many of these entries that come to work in my lab, and I think this will hopefully begin to get larger in the years to come. The last slide, I'd like to point out that cryo-EM is a broad discipline, and my own interest in this technology has been to bridge the gap, not just to look at proteins, but to look at viruses. Nearly half the work we've done in the last decade has been on HIV, leading to the first structure of the chimeric envelope glycoprotein, template for vaccine design. We've reported over 100 structures of HIV, influenza, and Ebola glycoproteins using a closely related method called electron tomography. We've looked at structures of whole cells, bacteria in the context of signal transduction, chemotaxis in particular. And finally, I haven't mentioned work we're doing to use these methods to look at very large structures, tissue in 3D, using a technology called focused ion beam imaging, which I originally seen in the semiconductor world, but now applied